Hey, hi, hello there, and welcome to yet another beautiful day in the Biology Bunker, where today we are jumping into Unit 2. All right, so you guys need your new Unit 2 packets. They are up on the front desk, which is right there. So you guys go and grab one of those. And what we are going to be working on today is the first section of it, which is dealing with the cell membrane. Now on this note packet, I even leveled up and tried to add in page numbers at the bottom of the page. So when you look in the instructions here, it even tells you the page number reference that you guys are going to be needing. So today you are going to be looking at pages 2 and 3. So the two that are dealing with the cell membrane, just one page front and back essentially, is what we're working on today. All right, so now let's skinny over to Google Classroom, and this is assignment number 37, okay? So right here, you've got the slides if you want to look at it that way, or you also have the PDF that is already filled in, which I suggest let's take a look at that one. Okay, so let's open that one up, and I'm going to, granted that it decides that it wants to function, open it up in Kami. And if you guys open yours up in Kami, then you will be able to see what I am about to highlight. Okay, so we have talked in passing about the cell, and you guys have, as a whole, pretty much said that you are not super familiar with the pieces and parts of the cell, and that maybe possibly you only remember the mitochondria does something with energy. So this may be a bunch of brand new content, but it is mucho importante that we understand all of the mechanics of what a cell has, because it will be relevant in every single piece of content that we cover, because everything is made of cells in biology, because it's, you know, the study of life. All right, so the first of that is going to be dealing with the cell membrane, which hopefully you remember is the outside of the cell. Okay, so it is very graphic heavy, which I think is going to be extremely helpful for you guys as well. All right, so Kami has decided that it wants to join the party, so let's take a look here. All right, so right below here it says, cell membrane, label the different components. Okay, absolutely 1,000,000,000% we need to do that. All right, right here it mentions that there is the difference in the plant cell and the animal cell. In a couple more pages, we'll get more into that about what the differences are. As you can see, they are obviously different shapes, and they do have some different components in them. So most definitely we are going to need to label that and that right there, okay? And if you do not remember from 2 years ago, the plant cell also has the cell wall. So on the outside of the cell membrane, the plant also has the cell wall, as where the animal cell just has the cell membrane. Alrighty, then below that, this whole entire graphic is going to be extremely important, and there is going to be a lot of content that we cover in labs, and then also just application about like how cells work and why things do the things that they do or why they don't do the things that they do are going to be based off of this graphic. So every single thing you see here that is labeled, let's actually take time, effort, and energy to label them, okay? So right here you have a glycolipid, which is glycogen, and lipid, which starch, fat. We have our carbohydrate chains, like I said, glycogen, carb. All right, glycoprotein, glycogen, and a protein. Then we have some proteins. Integral proteins are called integral because they are actually included into this cell membrane. Then we've got some receptors. And then we have a receptor protein.
Please. Final call for any senior that has not had their cap and gown picture made in the choral music room. Final call for any senior that has not had their cap and gown picture made to the choral music room.
All right, you guys heard that. All right, and then we have the receptor protein, which is going to be what is poking out there for it to be picked up so that the signal is picked up. All right, then coming around here, we have the phospholipid bilayer. Okay, this is going to be extremely important for you guys to understand, because phospholipid bilayer means that it is 2 layers. If you remember back last unit, we talked about hydrophobic and hydrophilic. Okay, so the inside of this cell membrane is hydrophobic. It's scared of water. And the outside is hydrophilic. It loves water. So this little shape right here that you see, remember back to last unit where I said it was going to be important, that here we are again. All right, and I think that's what that's saying right there, that it's hydrophobic on the inside, hydrophilic on the outside. Alrighty, and then let's scoot this down just a little bit so that we can see a little bit more. Alrighty, then we've got here, opposite of the integral protein that is inside of it, we have a surface protein, you know, because it's on the surface. Alrighty. You can also hear that termed as peripheral protein, just like you have your peripheral vision, but we'll just say surface protein. We have a channel protein. Why? Because it forms a channel for things to go into the cell or out of the cell. We have a carrier protein that just provides a little bit of oomph for something to get into the cell. And then we've got a peripheral protein, and then we do also have cholesterol. Alrighty, so I highly suggest that you spend a decent amount of time, effort, and energy on labeling those accurately, and then also maybe color code them and have a couple of different colors that are listed, because brain studies show colors matter. Okay, so now let's take a look down here with talking about cholesterol. We've talked about cholesterol before, and we talked about your good cholesterol versus your bad cholesterol, your good fats versus your bad fats. So here we're picking up that convo again. It says it's a type of a steroid. It regulates the fluidity, the fluidness of the cell membrane. So at low temperatures, it keeps them spread out. So that is going to be important. How much of that I would write? That is up to you. How much of that conversation do you remember that we've already had? If you remember, the good fats are going to be liquid at room temperature. The bad fats are going to be solid at room temperature. Like, let's see, this morning I cooked bacon, and the bacon fat, as soon as it started cooling down, what did it do? Turned into a solid, because it is a more bad fat. So if we're talking about an animal cell, here we are, because bacon comes from a oink oink pig, and we're talking about animal cells. Alrighty, it once again draws out that phospholipid that you've already drawn before. So if you remember that super duper well, you may just say hydrophilic, hydrophobic, and draw a line. You may need to remind yourself, this is the head, this is the tail, even though it has two tails, so technically it's tails. Alrighty, and then we have the fluid mosaic model. Okay, so essentially that is just explaining the fact that it is fluid. It says phospholipids are able to move around each other like a fluid, because an animal moves. I'm an animal and I'm moving right now, and the way that I move all kind of sort of boils down to my cell membranes, and the fact that my cell membranes are allowing all of that movement because they are by nature fluid. Alrighty, so I would say able to move around, to have different kinds of proteins embedded within the bilayer. There you go. All right. So that is a very quick 10-minute long look at the first page. I hope that that was very straightforward for you. So let's take a brain break and then look at the second page.